So I'm going to speak to you not about DNA sequencing technology, which is what I normally speak about, but rather about the use of DNA sequencing technology that we did through our collaborators, the University Medical Center in Nijmegen, and present some results of some very interesting work that they've done in the world of diagnosing intellectual disability using whole genome sequencing. So first, some definitions. The definition of intellectual disability, so significant limitations in both intellectual function and adaptive behavior for kids, for people under 18 years of age. This affects about 0.5% of the population. You may see other bigger numbers around neurodegenerative diseases, but this is really a specific category called intellectual disability. There's certainly some known causes, but well over half of the cases have no known cause at all. And for the most part, parents don't exhibit the disease. So it kind of begs the question, could this be de novo mutations, which seems unlikely, right? There are very few de novo mutations, about 100, per generation. Could it really be de novo mutations causing this? And it takes very high quality whole human genome sequencing to go and answer that question, which is one of the things the team here set out to do. Now, recently there have been some really good pieces of work done applying exome sequencing to kind of the broad class of neurodevelopmental disorders. Francis Collins has a great blog, and he published on his blog back in December summary results of 3 of the studies, which I'm showing here on this slide. And what they show right over here is, I think the screen's too bright for that. The first of the studies, which is a kind of a small sample size, about 119, showed that about a 45% diagnostic yield. So in 45% of the cases that were unresolved by other methods, they were able to diagnose the genetic cause of intellectual disability. The 2 other cases, the Yang and the Lee cases, 25% and 31%. Larger sample sizes, probably slightly more accurate numbers, but still very, very encouraging results, in particular to the parents of these kids that suffer from these intellectual disabilities. Now, the study that we were involved in actually spans a little more technology scope because we had array technology on a set of kids, and then we took the negative by arrays and did exome sequencing on those, and then we took the negative by exome and did genome sequencing on those. So you get to see this cascade of 3 technologies where each next technology tries to diagnose the cases that the previous technology missed. And what you see by those numbers is that the arrays got 12%, exomes got 27%, genomes got 42%. There's some overlap there. If you combine all that data together, what you get is this. 1,500 patients go into the hopper. 62% of these intellectual disability kids can be diagnosed using whole genome sequencing technology. That is a dramatic result. It's a brand new result that's just come out of the ability to be able to sequence kids at high quality and go in and figure out which mutations are causing the difficulties. And if you see the pie chart on the right, what you see, another dramatic result, completely unexpected, was that of those 62% of cases that were diagnosed, 60% of them were caused by de novo mutations. Only 100 de novo mutations per generation, and yet that accounted for 60% of the intellectual disability cases. And still, an unfortunate 38%, there was no diagnosis even with whole human genome sequencing, which I'll speak to a little bit. So let me draw 3 conclusions from this. Conclusion number 1 is this is clinically actionable. We're moving this now from the world of research into the world of clinical action in 2 ways. First of all, we can end the diagnostic odyssey. The parents go through these enormously expensive and time-consuming process of specialist to specialist to try to understand an intellectual disability. In 62% of the cases, we can find the causal mutation and say, here it is. You don't need to keep asking what it is. Now we can figure out if there's anything to do about it. And in some cases, certainly not all, but in some cases, you can actually take specific actions to prevent further complications. In particular, a ketogenic diet for patients with a specific mutation, the PDHA1 mutation, which is an anti-epileptic pathway, and then also just doing anti-epileptic treatments and avoiding sodium channel blockers for another specific mutation. So when you know the specific mutation, you can actually take affirmative action to prevent bad things from happening. And in a lot of circumstances, you can do this at a much earlier age than when the disease actually presents itself, when you typically can't figure it out before the age of 3 or 4. Clinical action action number 2 is basic research. What about that 38%? You know, this is whole human genome sequencing. It's really high quality, it's really high coverage, and it misses more than a third of the cases. Well, the key in the paper that was published, the Gillison paper, is, the quote is, we found nothing we can interpret outside of the coding regions. So even though this is whole human genome sequencing, the physicians only look in the coding regions because that's the only place where there's good science to be able to do interpretation. You go outside the coding regions and no one knows what's there. You know, the ENCODE study recently said somewhere between 8 and 80% of the genome is involved in regulation, and nobody knows what's in that regulation area. We need a big push in basic research outside of the exome, and you know, that's our key unmet research need right now. We need more funding, and we need big data. One of the themes of this conference is big data. 1,000 genomes won't solve this. 100,000 genomes won't solve this. We need millions of genomes available for physicians with good phenotyping to really begin to understand the control regions of the genome. The last conclusion is around reproductive health. First of all, think about our public policy in prenatal testing. We pay for Down syndrome testing as a society, as a global society, but the risk of Down syndrome testing is about 1 in 1,000. The risk of all these other intellectual disabilities is about 5 in 1,000. Okay, so 5 times as many kids have that intellectual disability than Down syndrome. What is the price of this diagnostic, a whole human genome diagnostic, before public and private healthcare systems say it's more cost effective to test everybody than to pay for the 5 in 1,000 that have the intellectual disability for all of their lives? We don't know exactly what that price is, but we're getting close. So when that price becomes low enough, we'll see a whole revolution in testing for intellectual disability that just follows in the footsteps of the Down syndrome testing that's been going on for decades throughout the world. And then the final conclusion, which is kind of fun, is this is all being caused by de novo mutation. The sad part of that is you cannot prevent de novo mutations. It cannot be done. But we can take some action. You know, the one known risk factor for de novo mutation is the age of the father at conception. And my apologies for the small data on the lower right-hand side of the slide that you can't see, but I'll tell you what it says. What it says is at the age of 20, at the father's age of conception, the number of de novo mutations is around 40. And by the time the father's age doubles from 20 to 40, the number of de novo mutations in the sperm doubles from 40 to 80. That's a 2x risk factor in de novo mutation. Now, assuming they're uniformly distributed, which they're probably not, we just don't know that yet, still it's potentially a factor of 2 in risk of having a kid with intellectual disability just by having the sperms go through fewer cycles of reproduction and accumulate fewer DNA errors as they do that. So it begs the public policy question, should boys freeze their sperms at the age of 17 and then for the rest of their lives use that low mutated sperm for having more healthy children? So I leave that public policy question to the public policy makers in the room, and that would be a big change to our society, shall we say.